really only eat about 130. So between two days, would you like another last one there whenever you want another last turn?
I already asked one.
Hello.
Hello.
Hi, how's it going, everyone? I don't think I really need this. It's a good sized crowd tonight. I'm Monica, and I'm a representative from the Women in Science Engineering up at UCSC, where we put on, in collaboration with the Crate Place, Science on Tap. And so this evening we have Professor Angela Brooks, and she will be talking a little bit about her research, and her talk tonight is Mining Genomes to Discover New Targeted Therapies. And a little bit about Angela. She is a true Californian. She's from Riverside, went and got her bachelor's at UC San Diego.
Yep.
Right.
Which college?
Rafael.
Oh, okay, that's okay, Marshall. That's
She did a postdoc at the Dana-Farber Cancer Institute, and now she is here and then she did her PhD up at Berkeley, and then came back a little bit down the coast, and is now an assistant professor in biomolecular engineering, and also a faculty at the Genomics Institute. And so I would like to welcome Angela and thank her for speaking tonight. Thank the Crate Place for hosting us as well. Please, I guess, work with your servers as they navigate the floor, and let's welcome Angela.
Thanks for coming. I don't know if can everyone hear me okay? Do you want a little louder?
A little more?
I'll also try to speak a little louder too. Thanks again, everyone, for coming. So I'll start off. We'll try the video. See if it works. Okay. So we'll see if the video works. If it doesn't, I can wing it from there. But I wanted to show this video because actually my first experience, or my first time learning about targeted therapies, was actually due to a postdoc that I worked with while I was in graduate school. Her name is Maki Nada. And shortly after leaving the lab, she was actually diagnosed with lung adenocarcinoma. And her story was actually featured when Francis Collins, who's the director of the NIH, gave a speech. Oh yeah, thank you. Yeah. Okay, now. So Francis Collins gave a speech basically asking for funding towards these types of research for precision medicine, and Maki's story was used as an example. So we'll see if this video works.
Maki Nada was diagnosed with stage 3B adenocarcinoma of the lung in 2008. It was completely unexpected. She was just 36 years old. Never. Her tumor was very large, 7 centimeters, with a very low likelihood of survival beyond the origin. Maki began and recommended standard chemotherapy. Her doctor suspected she might have a particular mutation in a gene called epidermal growth factor receptor, EGFR. Genetic testing confirmed their hunch, and Maki was prescribed Tarceva, a drug that precisely blocks EGFR's signal. After 3 months of treatment, Maki's large tumor shrunk dramatically. This was followed by surgery to remove cancerous tissue plus retreatment with Tarceva. Today, 7 years after her diagnosis, her doctors can detect no signs of cancer. Once more, during the extra time provided by this approach, Maki competed a triathlon, landed her dream job as a biology professor at Ithaca College, and welcomed a healthy baby girl. Clearly, we need many more stories like Maki's. That is our dream, and I'm sure it's yours too. With your support,
So,
this initiative will focus on cancer.
okay. So it is, really, this was my first experience learning about targeted therapies, and this was before joining the lab of Matthew Meyerson when I was a postdoc. and actually didn't even realize until I joined the lab that his research actually led to the identification that these certain mutations in EGFR conferred sensitivity to certain drugs. And so it was so her story and my research is kind of all ties together, and it's really interesting to think about the impact that our research can actually make on people's lives. So I want to talk about a little bit about Maki's story as a way to start off about how targeted therapy works. So actually now it's a clinical example where if a patient comes in with lung adenocarcinoma, they're sequenced for EGFR mutations, and then subsequently, if they have a particular type of EGFR mutation, it's already known that there's a particular inhibitor that those patients can receive, and they respond very well. And actually, it was shown that if they were given normal standard chemotherapy, they actually responded worse than people that didn't have a mutation. So it's actually really important for a particular subset of patients that have lung cancer to do this type of genetic test. So how does that actually work? So, why does precision medicine work? So to kind of step through that, I'll go over sort of the central dogma of molecular biology. So for those of you who are graduate students here, you could probably present some of these slides, but I'll just go over the overview. So how is information encoded in DNA? So here is a picture of a cell. The cell has a nucleus. Inside the nucleus, there are chromosomes. And what chromosomes are is a structure that our DNA or encoded information is wrapped in. The DNA consists of a helical structure of nucleotides that base pair together, and that's the basis of the genetic code. And so what does that code actually do? What maybe most of you learned maybe in basic biology is that again, there's DNA that's double-stranded, has particular base sequences. Those base sequences at the DNA level are then transcribed or copied into what's called mRNA. So you get a copy of one of those strands of DNA. And then that mRNA stands for messenger RNA. It then gets translated into a protein. And so there's a genetic code which involves three bases of nucleotides for every amino acid. And then when that protein is produced, that protein has some biological function. So what happens in cancer? So in cancer, again, in normal cells, you have, again, this DNA to mRNA to protein. And typically in cancer cells, you get a mutation, so potentially a point mutation that changes the sequence at a particular position. And so since that sequence changes, that then gets transcribed or copied into mRNA, and that sequence, that error sequence is then kept in the mRNA, and then that translates into a different amino acid sequence. So instead of a tyrosine, which occurred in the normal protein, you get now, say, a serine. And so what happens frequently, or what we think is happening with cancer cells, is many of these mutations occur in a cell, and it gives a selective advantage, and you get uncontrollable growth. So how targeted therapies work typically is that there's some drug or inhibitor, could be a small molecule, it could be an antibody, but it's specific for the mutated version. And I'm just diagramming that here as like a red box that kind of fits with this mutated amino acid sequence, whereas that inhibitor doesn't quite recognize the wild type version of the protein. So you can the ideal targeted therapy is something that won't kill off or kill off your normal cells, but something that's specific for your mutated cells. And this is essentially how the EGFR inhibitor works. So really the goal of precision medicine is then to find additional examples of this, where you have a cancer patient that then gets their genome sequenced or set of genes sequenced, you can identify the gene mutation and then decide what targeted therapy could work for them. And so to figure out other cases of this, one approach is to do cancer genomics, which is essentially sequence all possible tumors that come through clinics to see what are all the mutations that are occurring in existing cancer patients. And if we can identify those specific mutations, we may, for certain patients, we'll know, excuse me, if there are mutations that are targetable. So this example, I mentioned the EGFR mutation. BRAF is another gene that has a targetable mutation. But our goal of doing these cancer genomic studies, or one of the goals, is to identify potentially new novel targetable mutations that haven't been looked at previously. So what do we do for cancer genomics? So for an individual patient, so here I'm just diagramming a patient with lung cancer, you can take an actual biopsy from their tumor cells and sequence the DNA of the tumor cells. And then also a typical standard for cancer genome sequencing is also to sequence normal DNA, and so typically that is in the blood. So for example, for lung cancer, the tumor cells are thought to be localized right at the site of the tumor, and then all the rest of your body is normal, so you can just take an easy blood sample that's not invasive. So what we can do is then take the DNA from the normal and tumor tissue, and then sequence the DNA. And the way most of these sequencing technologies work is you take the DNA and fragment it into smaller pieces. And so what I'm diagramming here is each of these gray bars represents one of those small segments of DNA that was sequenced from the patient. And those segments were then matched up to a human genome reference to know what gene that little sequence came from. And so what we do in cancer genomic studies is look at positions where there's, and I hope you guys can kind of see, at least there's a little coloring. So each DNA segment that was sequenced, what is highlighted within that, what's called a sequenced read, what is highlighted is a sequence that differs from the genome reference. And so what you can see for this case, there's a position that's only found to be different from the human reference in the tumor tissue DNA, but not in the normal. And so that would represent a sequence that was only found in the cancer cells, and would then be a candidate mutation that was potentially causing that cell to become cancerous. So what we do is we do this sequencing for individual patients, but we do that across many patients with the same tumor or different tumors. And the idea is we want to find genes that are frequently mutated across many tumors to highlight specific genes that are commonly altered. So there's a couple of reasons why that's interesting. So one is you can understand the biology. So if the same gene is mutated all the time, it must be doing something to the cell to cause cancer. And then the other reason why we would want to find recurrent mutations is also for targeted therapy. So if you could find a drug that impacts half of all cancers, and that is a potentially good type of target to follow up on. So in my postdoc work, I was involved in this study where we did this for lung cancer, and we did sequencing of 230 tumors. And out of that, we found, we identified genes that were similar to EGFR. So we knew that EGFR is a commonly mutated gene in lung cancer, and we also knew that that was a targetable mutation. So this is a pie chart from that paper. It's a report of our results. And we saw that about 11% of those patients had EGFR mutations. And also, the most frequent one was KRAS, which was previously known before. Unfortunately, KRAS is not a targetable mutation. People are trying to develop therapies for KRAS, obviously because it's so frequent, but it's a challenging gene to target. But the focus of my work and my contribution to this work was to look at this particular slice of the pie. I'm highlighting what's called MET EX14, and so EX stands for exon 14, and that occurred in about 4% of cancers. So now I'll go into, well, why does it have that little extra label? What is X14 or exon 14? So now I'm going back to actually what my graduate work was about, and to look at the way genes are regulated. And one of the ways that we think genes are regulated is through this process called alternative splicing. Back when the human genome was sequenced, other organisms had been sequenced. So what I'm showing here is kind of the result of genome sequencing of worm, flies, fish, and humans. And when we finally sequenced the human genome, everyone was kind of sad to see that the number of genes that we have as humans is not that much different than the number of genes that worms have. So we thought that can't be right. We definitely look different than worms. So, maybe there's other things going on there. So one of the components that we think contributes to our complexity compared to, say, a worm or a fly, is through how genes are regulated. And as I mentioned before, one of the processes I study is how genes produce mRNA. And so now what I'm showing is if you count the number of mRNAs that come from each of these genes, you definitely see an increase or more mRNAs observed, more unique mRNAs observed in humans compared to the other organisms. And so I'll talk about how does this actually happen and how this relates to cancer. So I mentioned, so a given gene is a particular segment of the genome. So I described this long DNA that has all the nucleotides, nucleotide information. So a gene is sort of a specific segment of the whole genome, and it's a unit of information. And what I'm representing, the gene are these dark gray boxes, and in between there's what's considered non-coding or portions of the gene that aren't being produced or aren't being used in the eventual mRNA. So as I mentioned before, you have the DNA and that gets copied into mRNA. And so actually there's this pre-mRNA step. So the first copy of DNA to RNA is essentially exactly the same copy of the DNA, the gene level. Here I'm highlighting again, the boxes are portions that end up in the final mRNA, and then these smaller lines are called introns, and they're typically removed. So what alternative splicing allows you to do is from one primary message, you can make many different combinations as an output. So this is an example where you can have you keep all of the boxes that are diagrammed here, and say that is an expressed mRNA or a messenger RNA used in lung. But maybe in skeletal muscle, you use a different combination of exons. So here I'm showing all the same exons being used, except skipping this blue exon. And then maybe you can have even more diversity where, say in brain, you have a different type of mRNA that's expressed. And so what this allows you to have is from one unit of information, you can have many different functions, and you can kind of fine-tune how genes are regulated. So I mentioned that one way is you can have kind of tissue specificity, so certain types of forms of the gene is expressed in different tissues. But you can also have different functions of the gene. So this long one has a completely different function than the second or third. So what's also done in many cancer genome studies is transcriptome sequencing. That's a long word for basically mRNA sequencing. So what we can actually do is ask what are all the mRNAs that are produced by the normal cells and the tumor cells? And so we can ask, are there different gene products that are being produced by the tumor cells? So here I'm showing kind of that same diagram where you have different mRNAs that were sequenced in the normal tissue, and then potentially the same mRNAs were observed in the tumor, but maybe you get a different set of transcripts or mRNAs that are seen in the tumor. So now we're going to go back to what this MET exon 14 is. So what we found in this large study looking at 230 lung cancers, we found these mutations that occurred in the DNA, but not in these protein coding regions. So if you remember back, I was talking about mutations that change the sequence, that changes the protein sequence. So this is actually a sequence that doesn't occur in those coding or protein, the part of the message that codes for protein. So many of these mutations in this MET gene occur kind of right near the coding sequence. And so what happens is then that gets copied into the pre-mRNA, and what it's actually mutating is a recognition site. So it's disrupting how this piece of mRNA is recognized to then be put into the final message. So what happens then is with that mutation, you lose the recognition that exon should be kept, and you get this what's called an oncogenic form of MET. So if you kind of look at the ordering of the potential exons that could be used, what I'm showing here is that middle orange exon is not being included into this final mRNA. And so this version of the MET gene is only found in tumors, and it has been shown to cause, so if you introduce this transcript into cells, it actually causes cells to look like cancer cells. And and again, what I want to stress is this is not a rare thing that happens. It does happen in about 4% of lung cancers, and lung cancer is one of the most common forms of cancer, and also one of the highest forms of cancer death. So by studying, even though it's only 4% of patients, it actually does impact many individuals. So this was actually my first time working on something, because before I actually worked in Drosophila genomics, which was awesome, and I learned a lot, but this is my first time working on a problem that actually impacted patients. So it was personally really great to work on. So later, after this paper was published, I think more clinicians and people, so I don't personally work on designing drugs or finding drugs. I kind of work on the basic science side, but I'd love to work with others that are on the more therapeutic side. So other people, after we published this paper, followed up and basically we highlight in this paper this is an important mutation to look at for patients, and afterwards. And now it's continued to be an interesting target to look at, and this paper was one of the first to show why it's an interesting target to look at. So what I'm showing here are two patients before and after treatment. So both of these patients had these MET splicing mutations, and this red arrow is showing the tumor in patient 4 and also patient 5. And then after they were given crizotinib, which was actually a type of targeted therapy for MET, but not for this specific mutation, so it wasn't clear that this specific mutation would respond to this therapy. So what this paper showed is indeed that patients that had this mutation would respond. So you can see a reduction in their tumors after treatment. And also, if you notice the month after they're treated, it's quite dramatic. So for patient 4, it's only a month after that you see that much shrinkage, and then for patient 5, it was about a month as well. So that was work that I did in my postdoc, and I just started my lab last July, and I actually have a couple lab members here, which is great. I'll acknowledge them again later, but so one of the goals of my lab, one of the main goals of my lab is actually to find new instances of MET exon 14, and how common does this occur, and how much are we missing by not kind of looking into this kind of more novel way that genes can be altered. And also what's very compelling is, again, here's that pie chart. If you notice that there was, I don't know if anyone saw that earlier, but there's about 24% of the lung cancer patients that had no identifiable mutation that could explain why those patients had lung cancers. Whereas, the other, 75% or so, we could find some gene mutation that made sense. And so we're kind of, so one aspect is we're kind of digging into this, into more detail into the patients that are in this category of none, to see if there's anything interesting or potential new targetable therapy there. So this is all, so the rest of the slides is all in progress work, unpublished and all that. So now TCGA has expanded their set of tumors that they have. So from our original paper, we only examined 230 tumors, and now all the tumors have been sequenced that are part of this cancer genome effort. So we now have about 500 lung tumors, so about twice as many samples as before. So maybe we can pick up more rare mutations that are occurring. So we have 500 tumors that were sequenced. And again, from each of those tumors, we have DNA sequencing and this RNA or mRNA sequencing. And so what we're digging for or mining in this data is finding cases where there's a DNA sequence that occurs, and in that same patient, you see some different looking mRNA. And so one of the promising candidates that's come out of this initial look is an exon in ERBB2. And so this is real data I'm showing. So when we did that, so we basically, again, looked at all these exons, looked for mutations nearby, and said if there's anything weird about the RNA. And we can quantify how much of an exon is existing in all the mRNAs that are produced, and we quantify this by asking what % of all mRNAs that are expressed in a given patient tumor has that exon. And so what I'm showing is the quantification of this exon 16 across all the lung cancer patients. And this looks like basically a straight line, but that's because pretty much all patients, or most patients, express that exon 16, and all mRNAs that come from those cells express exon 16. But then there's this one dot here, and that one dot is one patient that has a mutation right at that exon 16. And so this is really compelling evidence that that DNA level mutation is causing the RNA level change, so to skip that exon 16. So we actually see hundreds and hundreds of these cases, and so the reason why we were particularly focused on this ERBB2 was because ERBB2 is already a known targetable gene. So, and actually it's typically targetable in breast cancers. I think there's some indication that people that have mutations in this gene in lung cancer can respond to therapies, but it's been pretty well established that patients with breast cancer that have a different type of mutation, so not these splicing mutations, but other alterations in ERBB2 respond very well to certain targetable therapies. And so in terms of when you're selecting out genes to focus on, if there's already a targetable therapy for a gene, you know it has the potential to be targetable. And so even if this mutation doesn't quite so even if that therapy doesn't quite target this mutation, you know it's potentially possible. So it's a better target to go after than something that totally doesn't have a therapy. So this is why we're more keen on this mutation. Also, there's evidence that exon skipping is actually causing causing normal cells to be cancerous. And so now that we have this particular candidate that we want to look more into, and we also have, as I mentioned, hundreds of other candidates like this, we're using a new, and I guess new in the past 5 years or 4 to 5 years, we're using a new technology to actually experimentally ask, if you cause this mutation, does that cause normal cells to become cancerous cells? And so that new technology is called CRISPR-Cas9. I don't know, for those who are not graduate students, who has heard of this technology? I said those who are not. Oh, you're not graduate. Okay, not graduate students or associate. Okay. And actually, I purposefully wanted to, just talk about this aspect of my research because it's an amazing new biotechnology that I think everyone should learn about because it has a lot of implications beyond what I'm talking about. But so I just wanted to talk about how we're using it and why it's so powerful. Some basic scientists who are looking at bacteria realized that they had some weird defense mechanism against viruses. And after further investigating, they realized that these these bacteria, the certain class of bacteria, would express this RNA. So here that's diagrammed by this kind of thin black line. So these bacteria would express these guide RNAs that would target or match exactly to viral genome sequences. And so what that would do by targeting the viral genome sequence, the bacteria had this other protein called Cas9 that could then cut the DNA at that specific site. And so many researchers realized that this might be a powerful tool, even though this works in bacteria. Can we take these components and put them into human cells and do the same thing? And indeed, we can do that now. So we can take almost any genomic DNA sequence in cells and design an RNA that targets or base pairs exactly to a sequence in the genome, and then cut at a specific site. So this has huge implications for in lots of areas. But we're using this technology in order to test our splicing mutations. So what we're doing is, so here's again this diagram of a gene at the DNA level. And so we can use this CRISPR-Cas9 system to design an RNA that complements exactly that position that we're seeing in human tumors and cut right at that site and essentially mutate the same position that we're seeing that's mutated in a cancer patient. And so by causing that cut, we can, what happens is that, again, that sequence is mutated, and then it's copied into the pre-mRNA, and then what we'd expect then is that we get a final messenger RNA that lacks that exon to basically recapitulate exactly what we're observing in human patients, but we can do that in an experimental controlled setting. And then we can actually ask, ask, if you create this mutation, does that actually cause some functional impact or cause normal cells to turn into cancerous cells? And so what we're designing are these type of experiments where we take normal lung cells, and I quote them normal because pretty much any cell line that people use for experiments aren't actually normal. They're kind of modified a little bit so that they grow in dishes. So we're taking normal lung cells, and we add a control guide RNA, so something that complements that exon sequence we want to target, and then we also introduce into normal cells a guide RNA that targets that exon that we're interested in. And what we're trying to ask is, do we see a difference in the transformation of the cells? And the way we assay that, or one way that we're assaying that, is through this what's called a colony formation assay. And so when you plate these cells on dishes, if these cells are normal, they kind of don't form big clumps on the dish. But if you've somehow transformed them or they become cancerous cells, then they have uncontrolled cell growth in these colonies, and you can see them as big black dots or big dots on the dish. So this is kind of my conclusion slide. So thanks for your attention so far. So really what our lab is doing is mining these cancer genomes computationally, and actually I didn't really go into all those details. If people have questions about that, I can answer them. But we're taking existing cancer genome data and looking for cases where there's interesting mutations to follow up on. We're experimentally testing them, and hopefully can work with others that can then design or look into existing drugs that might work for those specific patients. So first I'd like to thank my lab. So I just started in July, and actually a couple of my lab members are here. So Cameron and Pratibha are here. And some of the work that I described was the project of Max, who's an undergrad, and Eva, who's a specialist in the lab. And then also a lot of this work was carried over from my postdoc work with Matthew Meyerson, and I worked with great colleagues back in my postdoc work, and I've been collaborating or working with other labs here at UCSC. And then finally, would really like to thank the cancer patients who donated their samples in order to do this research. The Cancer Genome Atlas is primarily the resource that we use for this data, and then additional funding is by the Damon Runyon Cancer Research Foundation. And this is just a plug if people are computationally inclined. Some of the analysis was done with a program that we developed called JunkBase. So that's the link to it. And that's all. So I can take your questions.
The cancer patients who donated, do you have any idea where they came from?
So the question was, for the cancer patients who donated their samples, do we know their location? And the answer is no. So I say that cautiously because you could probably infer it for maybe some subset, but what's really stressed in a lot of these cancer genome studies is making anonymous samples. This is actually a huge discussion about, what does it mean to let your genome be publicly available and know that that particular cancer patient is, me, and what does that mean for me? And I think some of the ethical considerations is, would I be treated differently? Like if suddenly if I was trying to keep it a secret that I had cancer, like would I lose my job if someone knew about that, or lose my insurance, more importantly? And so there's a lot of work to try to prevent stuff like that, but for now, pretty much all the cancer genome data is anonymized, and you can't identify a particular person. Yeah.
Is anyone taking some of these common mutations, translating them into proteins, and then figuring out what the structure is, so that you can see what the active sites would look like if you could grab onto them, antibodies or something?
Yeah, so the question was, are people taking these mutations and translating them and then putting them and then doing structural analysis to see if there's any, are those mutations in active sites, or could you target an antibody? And the answer is yes. So there are many groups that are, there's actually one group in particular that took known structures and just mapped. So many of the mutations happen to occur in just one position, and so it's kind of easy to take the normal protein structure and just find the location of the mutation. And we do see that, in the protein coding space, they cluster in certain positions that seem to maybe interact with other proteins. So people are looking into that. That's a great question. I honestly don't know the exact method of delivery. I don't think it's oral. It's probably through an IV that has a drug. But honestly, that's a great question. I don't know. Yeah.
Most of these targeted therapies are inhibitors, I'm assuming, right? How does the inhibitor, does the therapy have to enter the cell, enter the nucleus?
Yeah. So the next question was, how are these inhibitors? So many of them are inhibitors of proteins. So a lot of these targeted therapies, what happens in the normal, the mutations cause the protein to be hyperactive, and so that's why if you can inhibit that hyperactivity, that's why certain cancer cells become more susceptible to the therapy. And then your next question is, how does the actual drug get delivered to cells? So some of them are small molecules, meaning that they can potentially enter into the cell. Some of the therapies are antibody-based therapies. So if you have a cell surface protein, then those are more easily recognizable by what's called an antibody that can then find cells that have that particular mutation. And actually, the tissue site is an issue. So in the case of EGFR, like I talked about, EGFR is one of the targetable therapies. And actually in glioblastoma, which is a type of brain cancer, those type of mutations occur very frequently, but because we think the blood-brain barrier, it's hard to get those type of drugs to the brain. So if someone has glioblastoma and an EGFR mutation, we have a therapy for it, but it's hard to actually deliver it.
Do we have ways to get past the blood-brain barrier?
Do you want to work on that problem? I don't think so. My impression is no, but yeah, I think that's an active area of research. So there's a question back there. Yeah.
So with that group of like 290 or so tumor cells that had their genes, the gene sequence, there was that big group of none.
Yes.
Was that a group of tumor cells that didn't have noticeable like gene mutations?
So yeah, I can go over that. So those were samples that had. So those were samples that they had mutations. So that's and I didn't go into detail. Actually, particularly for lung cancer, it's one of those tumors that are highly mutated. So we actually see thousands and thousands. So for a given patient, we see thousands and thousands and thousands of mutations. And it so happens that one of those mutations occur in in particular genes where we know that that mutation causes. So there's different debates about how mutational processes work and how that creates cancer. But one model is depending on the mutation rate in the cell, you're sort of randomly mutating different parts of the genome, and if you happen to be unlucky and that one cell that gets a mutation and it happens in a what are considered driver genes, then that's why those are selected for, and those cancer cells then can have some selective advantage and then continue to grow. And so the genes that are listed, the 75% that has some gene attribute, we know that gene is sort of a known driver gene or has the potential to cause cancer. But in the none, it means they had a lot of mutations, but they didn't have any mutations in these well-studied genes. And so part of what, so they didn't have mutations in well-studied genes, or they didn't have the kind of normal mutation that people have experimentally looked at previously. And so that's why we're looking into this none category to see if there's anything that can be further looked into, and to explain, why were these 25%, having lung cancer. Yeah.
What does the color coding correspond to in this pie chart? It seems like they're genes that are grouped into different.
Yeah, that's a great question. So the reddish places are things that were really well characterized before, that were really known to be implicated in lung cancer as a driver gene. The blue are sort of newer. So from this genomic study, we found these newer examples of genes that had the potential to be drivers. And so that's why they're colored in blue. Kind of like new in this study. And then the green is like, we still don't know what's going on with those. Yeah.
So why did you pick exon 14?
Great question. So